Hi, everyone. My name is Rachel Detwiler. I'm a fourth-year BME grad student in the lab of Dr. Jessica Kramer. And today I'm going to be presenting some work titled Synthesis of Polyproline towards Engineering Mucus-Mimicking Hydrogels. I don't know how to change the slide. Okay, there we go. All right, sorry. So mucus is actually the first barrier that our body sees against pathogens in the outside world. It's the first layer of defense of our immune system. It's a substance that lines the epithelial surface of all of our body. It acts to lubricate and hydrate these surfaces, and it also houses the majority of the microbiome. Mucus is a hydrogel that's mostly composed of water and ions. But the primary gel forming component is actually 5 to 9 weight% high molecular weight glycoproteins called mucins. These mucins have large regions of proline and O-glycosylated threonine and serine, and this is termed the PTS domain. This area or this domain is important to the rigidity of the protein as well as structural diversity, and the glycosylation patterns here likely play roles in binding of bacteria, small molecules, and other proteins. The PTS domain exhibits what's known as a polyproline type 2 helical secondary structure, but actually polyproline itself can take on two different structures. The one shown on the left is the PP1. This is all cis isoforms of the amide bond. This happens in organic solvents, like the way that we actually make our materials. And then in aqueous environments, the polyproline takes on the structure of the PP2, trans isoforms of the amide bonds, and a more extended structure. This is thought to be very important to the structure of mucins as well as other proteins like collagen. So in our lab, the Kramer lab, we make glycosylated polypeptides as mimics for natural glycoproteins. To do this, we cyclize amino acids to form N-carboxyanhydrides or NCAs, and then we polymerize these NCAs with transition metal catalysts. This is a ring opening polymerization that forms a natural linkage between the peptides, and the transition metal initiators developed induce very rapid polymerization. The materials we make can be 10 to 900 amino acids in length. And the content of these polypeptides is tunable. This method is something that's well characterized for most canonical amino acids, but there is one that's a little bit different, and that is proline. So proline is actually the only natural amino acid. Sorry, I'm going to pull up my laser pointer. It's the only natural amino acid with a secondary amine. That's this right here. And that comes from the side chain on the amino acid wrapping back around and being covalently bound to that amine. This is important because when we go to make the NCAs shown here with these, there is no proton on this amine like there is in every other amino acid. So in past work, proline has actually been made into an NCA, but no one has ever successfully employed a transition metal initiator to make polyproline materials. So once these transition metal initiators were developed about 25 years ago, the group that created these did some very thorough NMR studies to show us each step with isotopic labeling and quenching. And through that, what's important to note is that there is a step that does require an amide proton on the monomers. And then the overall result of initiation is to create this metal complex with an aminoamidate. In that same study, they proposed a propagation mechanism. This is something that's really hard to study to capture these intermediates. But despite this, this is something that has been very well accepted as the mechanism without a whole lot of proof. So again, important to note that this also requires the amide proton, something that proline lacks. Around the same time, people created these nickel aminoamidate complexes to create functionalized polypeptides, where you could actually add any N group to the N terminus of your polypeptide material. So for this, we actually selected an azide so that we could make the end of our polymers clickable. So the overall goal here is that we desire to improve mucin scaffolds by engineering all the different components of the PTS domain. Work previous in our lab actually has already tackled the threonine and serine components, but what remained was to figure out how we could use proline in our research strategy. And our hypothesis is that we can actually use the nickel aminoamidate initiators that I mentioned to polymerize proline NCAs. So to start, we make the monomer here. It's actually very simple. We have Bach protected amino acid commercially available to us. Then in a pretty simple and scalable cyclization reaction, we're able to make our NCAs. Synthesis and purification of this can all be done within a day, which is awesome. And to kind of look at proving what has already been shown in the past, indeed, with the transition metal catalysts, when you introduce them to the proline NCAs, you do not form polypeptide. However, we do have successful polymerization when we subject them to the nickel aminoamidate complex. So towards this, we made several different polyprolines, and we're able to actually have these complete at a monomer and to initiate a ratio of up to 200 in less than an hour. This is very comparable to when you polymerize other NCAs with the traditional transition metal catalysts. And this is very fast compared to like the other techniques that have been used for proline NCA in the past. So on the left, I'm showing you the infrared spectra. You see the disappearance of the NCA anhydride peaks that are in the red here. The 1 hour time point just has now the formed amide bond in blue. On the right, this is a plot that shows the linear relationship between molecular weight and the monomer to initiator ratio. And this proves that we have tunable and controlled polymerization. The next thing that we wanted to look at, since mucins are actually multi-domain proteins, is we wanted to see if we could make block copolymers. So just like before, we take the proline NCA, we add the nickel initiator, we see that this reaction is completed by IR, and then we actually add back in a solution of another NCA. And again, once that's finished by IR, we have a diblock copolymer. The data, the plot on the right there is showing you guys a GPC LS trace with elution times for both the first block in green and then the diblock copolymer in purple. We use size exclusion chromatography. So with this, you would expect larger polymers to elute first, and that is indeed what we see here. And then one of the last things that we wanted to look at for this project, since the PTS domain is a polyproline type 2 helix, is we want to see what the transition of our polyproline materials looks like. Since we synthesize it in organic solvent, it takes on the PP1 more constricted structure. And so when you incubate it in water, we're interested in what this looks like, since we desire the transition to the polyproline type 2 helix. I'm showing you CD data on the right, and you can see that in green at time 0, you have what looks more like a polyproline type 1 helix, and then somewhere between 3 days and a week, it is fully transitioned to the polyproline type 2 that we desire. So we're really excited about the behavior of these materials in an aqueous system. So in conclusion, in this work, we proved that the nickel aminoamidate initiator can be employed to initiate and catalyze proline NCA polymerization for the first time. We're able to quite easily make end-functional, high molecular weight and tunable polyproline homo and co-polypeptides. And indeed, our materials do take on the PP1 structure in water, just as the PTS domains do in nature. And I published this work last summer. So with that, I'd like to thank my mentor, Dr. Kramer, the peers in my lab, and our funding sources. And do we have any questions? And I guess I'll give you this back.